Hello and welcome to the Genetics Module 6 exam video on positive repressible systems. So to start out, what is transcriptional regulation? It is when a gene or operon uses effector molecules, promoters, and or repressors to help control the amount of transcription occurs. This is usually done through allosteric feedback or binding sites which inhibit or promote the transcription. Most common form of regulation for gene expression within prokaryotes is this allosteric feedback. So we will be talking about positive repressible systems where the activator works to bring in DNA polymerase and help stimulate transcription within the operon. However, that product that is produced, which is normally a protein, acts as a small effector molecule and binds to the activator protein, changing the conformation so that transcription is no longer occurring and is now repressed because no DNA polymerase is coming through. So these are just some of the key players. The promoter helps to attract DNA polymerase. The activator protein binds to the promoter region to help stimulate polymerase activity and help bring in that DNA or RNA polymerase. The activator DNA binding site is where this activator protein binds to the gene. It is usually upstream of the promoter region. The small effector molecule, it binds to the activator and changes the conformation, so it's usually one of the proteins made from the structural genes. The structural genes are what is transcribed, translated, and made into a functional protein. The operator is where consensus sequences and those can be found, which help direct transcription, kind of tell things where to go. And then DNA polymerase transcribes the gene or DNA into mRNA. And if this were an RNA strand, you would use RNA polymerase. So how does a small molecule change things? In the presence, the effector molecule binds to the activator protein and changes its shape through allosteric inhibition so that it's no longer able to bind to DNA, and it turns the system off by making it so that the DNA or RNA polymerase does not want to bind to the strand. In the absence of it, the activator protein remains in the active conformation and will help promote the binding of DNA or RNA polymerase and the following transcription of the gene. Analogy. So when you're eating when hungry. So when you're hungry, your empty stomach acts as the activator protein that is binding to the promoter, helping to bring in signals to tell the person to eat and make sure that the movement of the hands bringing food to mouth is being transcribed. Once it is transcribed and you have eaten your fill, i.e. a whole burger and fries, the food acts as an effector molecule to change the conformation of the stomach. Now the stomach is full and no longer can send signals or bind to the brain to let transcribe of eating. So now you don't need to eat, and so you don't need those genes to be expressed. These are my references. Thank you so much.